Good afternoon. My name is David Egan. I'm the CEO of Core Life Analytics. Core Life Analytics is a company that helps biologists to analyze their own complex data. Now, I'd like to thank the organizers for inviting me to speak here, ODI Leeds and the MC of the Netherlands. But the first thing I have to start with actually is an abject apology, because I realized that in the 3.5 years or so that Core Life Analytics has been active, I've never actually had a business trip to the north of England. They've all been either to the obvious Oxford, Cambridge, London triangle in the south, or Scotland in the north. So yeah, I realize there is some very good work going on in the north, and I promise this is something that will be addressed when we're allowed to travel again. My interest in open data stems from the fact that during my postdoc in California back in the late 90s, I got interested in a relatively data-intensive area of biology. And this was in the use of automation for biology. And so what we were doing in the lab I was working in California is we're using robotic systems for setting up experiments in plates like this. And so this is a 384 well plate, and what you can see is that you can set up an individual experiment in each of these plates. And so this is technology that is used in heavily used in industry and also in academia. And this use of this technology turned into a career in what's known as high throughput screening, where I actually ended up delivering these automation services to groups in industry and in academic centers. My last position before founding Core Life Analytics was as the manager of a screening facility called the Cell Screening Core at the University Medical Center in Utrecht. There we had a robotic system for carrying out high throughput screening assays and microplates, but what we also had was an automated microscope. Essentially, this is just a microscope in a box that allows you to take images of cells in those 96 and 384 well plates that I described. Now, the automated microscopy gives you access to a technology called high content screening. And this just describes how it works. Basically, you can take images of fluorescently labeled cells using your automated microscope. And then with automated image analysis software that generally comes packaged with the instrument or open source software, you can extract numeric descriptors of the cells that you've been taking images of. So for example, if we look at this nucleus, we could extract numbers that describe the area, the shape, the perimeter, the intensity of the nucleus. Similarly, with the cell, we can identify shape, we can identify intensity of various labels, fibers, etc., spots. And of course, all of these numbers create a profile that's essentially describing the actual morphology, what this cell looks like. The power of this technology is that that profile is actually related to the mechanism of action of some chemical or genetic reagent that you might be testing. And so how it's used is that large libraries or smaller numbers of chemicals or genetic reagents can be tested in these plates, and then you can generate profiles that are related to the activity of the chemicals. Now, one problem we had at the Cell Screening Core is that while we had all the technology required to generate the images and extract the numeric features from the images, we didn't have any tools in order to help our clients analyze the data. And so essentially they couldn't make full use of this technology and do what we call multi-parametric analysis. In order to solve this problem, I hired a graduate student, Veena Dumta, who subsequently became my co-founder at Core Life Analytics. And what we did was we built a web-based tool called HC Stratominer. And the idea of this tool was that it would allow biologists to upload their data from these high-content screening experiments, and then it would walk them through a data analytics workflow. So it would allow them to isolate the metadata, carry out variable selection, essentially throw out the garbage, then do QC on the data, things that are specific to these type of experiments in microwell plates, plate normalization, transforming the data, scaling the data, and then on to what's known as data reduction or dimensionality reduction. This would reduce the very large numbers of features to a smaller number of relevant scores. We could then use these to identify the outliers, based on these profiles of numbers. And so essentially these were the ones that looked different from something else or looked similar to something else. Then we can cluster these to separate out the different types of profiles. And then if we found something interesting, we could build machine learning models and then apply these to the whole data set. So essentially this meant that what we were doing was we were allowing biologists to analyze their own data by turning data science into data technology. Initially, we were able to test and validate HD Strataminer using data generated at the cell screening core, but we quickly found that we needed larger and more complex data sets in order to really push the envelope and to show the value of the platform. Fortunately, in the high content field and the image-based screening field, there are a number of good open data repositories available. So the first one is the IDR, the image data repository that was built by Jason Svedlow and his group in Dundee, and the other is the Broad Bioimage Benchmark Collection that was developed at the Broad Institute in Cambridge, Massachusetts. So let's have a quick look at those. The Broad Benchmark Collection is quite straightforward. Basically, the idea is that it is a collection of high-quality image sets that can be used for benchmarking, and most of them have some sort of ground truth built into them. And so here you can see the collection of data sets, and what you can do is if you open a particular data set, you can get introduction to the experiment that was done, a reference, and then the images and whatever metadata is available. But it's simply a repository. You can't view the data or browse the data. You simply get some information about it, and then you can download the images and download the metadata that's available. The IDR, on the other hand, is far more elaborate. This is gives not only is it a repository for many screens, it gives a lot more functionality as regards browsing the data. So here, for example, you can search for a particular data set, and then it opens up here, and then you can see on the left all of the individual plates. And when you click on one of these, the actual plate pops up, and you can see a kind of a plate based map of all the images. If you want to look at one of the images more closely, then you click on it, and then up pops this elaborate browser. This is really cool. You can turn on and off the various channels here. You can adjust the scaling. You can actually copy and paste the scaling so that you compare multiple images using the same scaling. It's all quite elaborate. And when you go back to the actual screen, you can get information about the metadata that's related to the well, the compound structure, for example, the compound name. Also, at a higher level of the screen, you can download. There are files of metadata here that you can download. It is also possible to download images from the repository, but it's not as simple as the broad repository. You need to interact with an API, but it is doable, and you can get some help online to figure out how to do that. One very interesting screen that was present in both the Broad collection and the IDR was based on a paper that was published by Neil Carher from the University of Edinburgh back in 2010. This was interesting to us because it was a small molecule high content screen where Neil and his group tested 102 drugs in 8 different concentrations against a number of different cell lines. And the images to the data from one of these cell lines, MCF7, was uploaded into the repositories. And so these were the various compounds that were tested, and what was of interest to us was that they were from a wide variety of different mechanisms of action, and these mechanisms of action were well annotated in the metadata, along with the concentration and the various structures of the chemicals themselves. So what we did was we collaborated with Johnny Sexton at the University of Michigan in the US, and Johnny's group downloaded the images and ran them through Cell Profiler. So Cell Profiler is, as I described earlier, an open source image analysis software, which is extremely powerful. And so then we were able to take the numeric data that Johnny extracted, and this was 479 individual numbers, and this was done at cell level, meaning we had 479 features for every cell in the data set. And so this rich, large data set we were able to run through Strataminer, and again, as I described earlier on, we were able to upload the data, identify the metadata, and then carry on through the process of normalization, transformation, and scaling until dimensionality reduction. And then at dimensionality reduction, we were able to reduce these hundreds of features down to 9 robust individual scores called principal components. And Strataminer helps the user to decide how many different scores they should calculate in the principal component analysis. Let's jump into the Strataminer platform. I can show you a little bit about how this works and then what we did after this dimensionality reduction step, because this starts to get very interesting from this point. So here we are in the Strataminer web-based platform, and here on the left what you can see is the workflow that I described earlier, and now we're at this step here, the dimensionality reduction. And so what I've done here already is I've processed the data, and we've taken 336 features, and then we've reduced them down to 9 different what we call principal components. And the platform makes it very easy to investigate these different components, and you can see here what sort of features are loading on the different components. And then these individual scores, you can see how different types of chemicals actually trigger different scores and are related to different principal components. And so this is essentially the biology coming out in the data reduction. Now, as you can see here, this is kind of an indication of how the larger number of hundreds of features are reduced to 9 individual scores. And then we can take these individual scores and then we can carry on and use them to actually do hit picking in the next step. So what we do is we hit save and continue. And then what we can do is, just to demonstrate how we do this hit picking, we can generate here a 3-dimensional plot using just 3 of these different of these 9 principal components. So let's have a look at this. Now, here we've all these different classes of wells. These are all of the wells in the data that we've processed here. And let's turn off these ones, the samples, and let's just look at some pretty obvious controls here. Okay. Now, all of these are color-coded, and so these negative controls here, the ones that we know nothing should happen, are labeled in red here. And then we have various different types of controls labeled in different colors. The green ones are positive controls, where we know there's going to be a phenotype. Now, if you look at these positive controls, you can see that they're far away from the negative controls, okay, in 3-dimensional space. Here we're just plotting 3 of the components. And that's actually how we define our essentially phenotypic hits. These are far away because they look different from the cells in these wells. And what we can actually do is we can actually measure the distance from the negative controls to each of these. And that's indicative of how different they look. Okay. And then that's what we do. We actually calculate a geometric, what's called a Euclidean distance for each well, and then that can determine whether it's actually a significant outlier. So let's do that. We'll select all of our components for this, what we call hit picking step, and then we're just going to calculate the distance for every well from the negative controls, and then what we get is a list of hits. Then all of our distances are visualized, and here we can see our negative controls and then our positive controls. Then these are all the samples that we're testing, and then we can see that there are a number of hits here, things that are significantly different from the negative controls. And then these are dumped into a list, and then we can sort this list, we can filter it, and so on. Now that we've isolated these hits, what we can actually do is we can go back to the original 9 principal component scores, those 9 reduced scores that we generated, and then we can do clustering based on those scores. And here you can see the clustering. And now this is the beauty of the high content analysis, because now what you can see is that the chemicals here in the hits are actually clustering based on their mechanism of action. So what we can see here are cytochalasin, latrunculin, and cytochalasin, latrunculin. These are all actin inhibitors. Farther down here, what we can see is Taxol, epothalone B. These are all microtubule compounds. And then over here, what we can see is compounds that are DNA damaging agents. And so this is the real high content paradigm where we can take images, extract numbers, and then isolate chemicals that are giving us an effect, and then we can separate them according to mechanism of action, all based on a cell-based assay. So this is just another look at that clustering, and you can see here, here's our DNA replication and damage agents that are clustering here, and then down here are actin inhibitors, and then our microtubule modifiers. And another thing we can do with this distance score that we used is that we can actually plot it against the concentration of the chemical that's being involved. And what happens is that then, as you get an increase in concentration, if it's a bioactive, you get this nice sigmoidal curve. This makes it very easy in order to identify chemicals that are giving a biological effect. So if we look at that a little bit more closely here with one chemical called docetaxel, this is a microtubule inhibitor, what you can see is that as that distance score increases, we see the phenotype coming out. Here is one with a low distance. These are very similar to the negatives, intermediate distance, and now here a high distance. And you can see this is a very strong phenotype. If we look at latrunculin, which is a chemical, a drug with a different type of mechanism of action, it affects actin. Then what you can see is also low, that looks very similar to negatives. Medium, you see the phenotype starting to come out. And then with a high distance score, you can see there's a very strong phenotype. But it's very obvious that this is a very different phenotype from docetaxel. And so this is why they cluster separately on that hierarchical clustering diagram. Then what we can do is, because we have data at cell resolution, we can actually build AI models. And this is another way of doing the analysis within StrataMiner. We can isolate interesting wells that we have found during our clustering analysis, and we can label those and then use them to build AI models. Here we've built a random forest machine learning model based on these different reagents. And then what we can do is we can ask the question, okay, what wells look very similar to either docetaxel, latrunculin, doxorubicin, or AZI? And then what we can see is actually they pull out wells that have a similar mechanism of action. One thing that this highlights, the use of the StrataMiner platform with open data, it highlights that what we can do is actually iterative data mining. So what we can do is we can load our open data that we've extracted from images, from the IDR or from the Broad collection, and then using StrataMiner we can extract new knowledge from that. Then this new knowledge can be turned into metadata that we can merge with our original data and then do another round of analysis. And so this is essentially iterative data mining. It's the same way that people iterate and do multiple experiments based on answers they find in experiments. We can do the same thing with the analysis of our data. One thing that our work with these open data also shows is the actual challenges of using the open data. In certain cases, you end up with data that is unusual or weird formats. Some of the files can be very large and can be hard to handle. Also, some of the metadata can be split between different files, and so this is a critical point where you have to merge them, you have to join them, and sometimes this can be a challenge. As you saw with our images, downloading the images is possible, but then you do need advanced tools in order to extract the features from the images. And of course, you need some tool like StrataMiner for a biologist to be able to get some useful information out of the numeric data. Another problem, of course, that has been addressed earlier is that there's a lack of standardization. Now, will it be possible for everything to be done in a completely standardized fashion? I don't think that's realistic, but one thing that can greatly help with this is proper tools that are flexible enough to allow people to pull in, data from various different sources. One thing you noticed as you browse the various data sets in the image repositories is that they're all quite different. Different types of biology, different cell systems, even different species. And so they're hard to compare like with like. Now, what if there was a standard assay that people could test their chemicals against and so compare different experiments? Well, there is a platform that might make that possible, and that's called cell painting. Now, cell painting is a fairly straightforward protocol that uses 5 different dyes that label different parts of the cell, 8 different cellular compartments. And what this generates is a very rich, high content assay that can be used to profile genetic reagents or various different chemicals. And then the idea is that if you build up a kind of a library of these profiles, maybe that would be useful for comparing and and clustering with new chemicals or hits from new screens. And possibly you could compare between different experiments. And so this really would make open data from high content experiments extremely useful. And there are a number of consortia who are actually working on this idea, generating these libraries of profiles using cell painting. There's also a lot of interest in cell painting for things like predictive toxicology, where you could generate profiles of unwanted phenotypes, and then you could use those to predict unwanted phenotypes in novel chemicals that are coming out of screens. So I hope that gives you an idea of how we've been using open data and the possibilities and challenges of using open data. And I'd like to thank you for your attention, and I'm happy to answer any of your questions.